Hello friends, antibodies are one of the most commonly used tools in life science and clinical research. In this video, we will see how specificities of these antibodies are validated by gene knockdown and gene knockout methods. This is the representative image of antibody specificity validation, and here the presence of intense signal is indicating the antibody validation. So we'll begin with brief overview of antibodies, how these antibodies are being secreted by organisms' immune system and their wide research applications. Then we will see how immune secretion of antibodies is harnessed for large-scale production and why we should go for antibody validation. Finally, we will see how siRNA gene silencing and CRISPR-Cas9 methods are being used for knockdown and knockout validation of antibodies. Antibodies, also known as immunoglobulins, are glycoproteins produced by organisms' immune system in response to foreign invaders, which are antigens. These may be microorganisms, viruses, nucleic acid sequences, or any chemical compounds. These are Y-shaped protein having two light chains and two heavy chains which differ in their amino acid sequence and length. They are linked to each other via disulfide bonds. Antibodies contains constant region which are common to all the antibodies produced by particular species and variable regions which are unique and specific to particular epitope on the antigen. They have uppermost Fab domain or fragment antigen binding domain and lowermost FC domain, which is fragment crystallization domain. The binding of antibody to antigen doesn't mean that it will bind to whole antigen surface. Rather, particular antigen binding site or paratope in the variable region of antibody will be binding to particular epitope, which is 5 to 6 amino acid sequence on the antigen surface, and there are so many epitopes on the antigen surface which will be binding to their respective paratopes on the variable region of antibody. Okay, now let's see how immune secretion of antibodies occurs. The first encounter of antigen to the organism's immune system will trigger the immune cells like macrophages and dendritic cells. These are antigen presenting cells which will capture the antigen, break it down and present the peptide epitope on their surface. It will trigger the B cell lymphocytes. These are antibody producing cells. The active B cells undergo somatic hypermutation on the variable region of the antibody. It will give rise to new antibody with unique paratope, which with high affinity can bind to specific epitope on the antigen. After sufficient production of the antibodies, these will be released on the bloodstream where they will execute the elimination of the antigen. The Fab domain will be interacting with the antigen, whereas FC domain will be interacting to the effector cells like macrophages. After successful elimination of the antigens, some B cell populations will remain on the bloodstream as a standby cells for any possible re-encounter of the antigens. These are memory B cells, whereas some B cells populations will migrate to the bone marrow or spleen, where they will exist for the lifelong lifespan of the organisms as antibody producing cells. These are long-lived plasma cells. It is the binding specificities of antibodies that will make them ideal reagent for their research applications in life science and clinical research. It infers that a unique antigen binding site or paratope on variable region of antibody will be specifically binding to unique epitope on the antigen. So it will support the precise detection of target antigen. In Western blot, the proteins are separated electrophoretically, transferred to appropriate absorbent membrane, and proteins are detected with the help of labeled antibodies. In immunohistochemistry and immunocytochemistry, The antibodies are used to evaluate the presence and location of the proteins in situ in tissues and cells respectively. Similarly, in flow cytometry, the antibodies are used to determine the status of cell population after their surface labeling and subsequent analysis by flow cytometer. In immunoprecipitation or pull-down assay, the antibodies are used to label and precipitate or pull down the desired antigens in the biological fluids or any aqueous solution. Similarly, in enzyme-linked immunosorbent assay, the antibodies are used for quantification of any soluble antigens in biological fluids or cell culture supernatant. The understanding of how organism's immune system secretes the antibodies can be used for the production of custom antibodies, the antibodies that can specifically bind to the target antigen. First of all, the lab organisms like mouse, goat, rabbit, these are immunized with any target antigen. So few weeks after the immunization, the active B cells starts secreting the antibodies specific to that antigen. So the secreted antibodies from active memory B cells are harvested from the antisera, that is whole blood excluding the blood cells and fibrinogens. Here, at least twice immunization of the live animals is preferable. The intention is to produce the active memory B cells that produces the pool of antibodies that with higher average affinity can bind with any particular epitope of the antigen. Here, the antigen may have several epitopes on their surface. So accordingly, multiple clones of B cells are activated, and multiple clones of active memory B cells will give rise to antibodies that can bind to several epitopes of the antigen. That means the secreted antibodies are polyclonal in nature. So there is always possibility of cross-reactivity that gives false positive result. That is, there may be the conformation of antigen that is in fact not present in the sample. Monoclonal antibodies, on the other hand, have higher specificity. That is, all the monoclonal antibodies with higher affinity will bind to any particular epitope of the antigen. Here, the production of monoclonal antibodies begins with the immunization of the antigen to the lab animals, preferably mouse, several times, and the active B cells are harvested from the spleens. Then these normal B cells, which are unable to proliferate for the longer time, these are fused with myeloma cells, which are cancerous B cells, and it gives hybridoma cells. And these hybridoma cells are allowed to grow in the selective media in order to exclude the unfused B cells and myeloma cells. Then each hybridoma cells are allowed to grow as clones, and each clones are screened for the antibody production, and based on desired monoclonal antibodies, the specific clones of hybridoma cells are selected, and such clones are allowed to grow separately on tissue culture media, and then monoclonal antibodies are periodically harvested and purified from the culture media. Despite their higher specificity, the production of monoclonal antibodies is very expensive as well as time consuming, because it needs the expense of several liters of culture media in order to support the periodic extraction of monoclonal antibodies. Such produced antibodies will be used widely in life science and clinical research, where their specificity and sensitivity cannot be compromised. We already have growing issues of cross-reactivity, which means there may be occurrence of antigen-antibody reaction apart from with desired immunogen. It will give false positive results and experimental irreproducibility. So the lack of consistent results will cause huge loss of resources, and it will give false diagnostic reports for clinicians. The only solution to assure antibody specificity is the proper validation by using appropriate positive and negative controls. The positive controls may be cells or tissues which have higher expression of the target proteins endogenously, or they produce high amount of target proteins upon any stimuli induction. Still, only the use of positive control may not exclude the doubt of cross-reactivity. The higher signal achieved in positive control may also be due to cross-reactivity. It is because the antibody raised against particular antigen may have higher affinity to bind with several antigens when there is structural similarities in their epitopes. So negative control should also be included, which can exclude the doubt of non-specific binding. And negative control may be the cases of either gene knockdown or gene knockout. Gene knockdown validation is based on RNA interference mechanism. The expression of target gene is effectively silenced or knocked down by selective inactivation of corresponding mRNA. It is mediated by small interfering RNAs or siRNAs. At the beginning, siRNA target site is selected. Then shRNAs, that is small hairpin RNAs, are designed, which is precursor forms of the siRNAs. Then it is followed by the vector design having two DNA oligonucleotides, which are sense and antisense strands. After successful transfection of the DNA vector to appropriate cell line, the cellular transcription machinery will transcribe the foreign DNA and give rise to shRNAs. Then the endonuclease enzyme dicer will cleave the loop portion, and remaining will be double-stranded siRNA. This siRNA will bind to the RISC complex, RNA induced silencing complex. Then here the two strands of siRNAs are separated, and RISC complex will remain associated to a single strand of siRNA that is complementary to the target mRNA. It will then leads to the cleavage of target mRNA, and subsequently there will be decreased expression or decreased accumulation of the target proteins. When we see the knockdown validation results, we will see the weak signal in knockdown cases. It is because there is decreased expression of the target proteins. So even with use of the antibodies, there will be weak signal. Whereas in control siRNA or scramble siRNA case, there is no gene silencing, so there is a presence of strong signal. In this negative control or scramble siRNA, it will also code for the same nucleotide sequence as siRNA, but the nucleotide sequences are scrambled or disarranged here and there so that there won't be occurring any gene silencing. Here the consistent bands refers to the housekeeping protein. It confirms that there is equal loading of the proteins in all the cases. The gene knockdown validation involves mRNA degradation, not the complete removal of the mRNA, so the effect is only transient. After successful transfection, the knockdown effects remain only for 5 to 7 days. In addition, the mRNAs may sometimes evade the RNAi interference mechanism. So this is the temporary effect. If we want to go for the stable negative control, the gene knockout methods may be preferable, where the genes are removed genome-wide for permanent basis. Gene knockout represents one of the most trusted and accepted negative control for the antibody validation. It uses CRISPR's guide RNA to direct the endonuclease Cas9 for sequence-specific identification of gene of interest, where this endonuclease Cas9 will cause double-stranded break around the target site, and it leads to the repair mechanism, which may be deletion, insertion, or frame shift mutation. And these repair mechanisms are error prone, that will disable the gene. So consequently, there is a removal of the gene genome wide, and there won't be any expression or accumulation of the target proteins. So when we go for the results, there won't be any signal in knockout case, whereas there will be specific signal in case of wild type cell lysates. So when we assess for the knockout validation results, what we see is complete absence of the signal from the knockout case, whereas there will be the presence of signal in wild type case. Say in this Western blot results, lane 1 is for wild type cell lysates, lane 2 is for knockout cell lysates. There is ubiquitous expression of the housekeeping protein beta-actin, represented by a red band. So there is equal loading of the protein in both cases, whereas the expected 34 kilodalton base is only present in case of wild type. It is completely absent from the knockout case. Here in immunocytochemistry result, the localization of P62 protein is only present in case of wild type HeLa case cell lysates, whereas it is the signal is completely absent from the knockout case. So that means even with use of the antibody, there won't be any antigen antibody reaction, and because there is complete absence of the target protein expression, therefore there won't be any signal. So it indicates the validation of antibody specificity. Despite being stable negative control, the knockout validation is not an unanimous solution. There may occur off-target effects, that is, there is possibility of other proteins elimination apart from the target proteins that will still give the space for cross-reactivity. How it happens? There is a sequence called PAM motif, that is protospacer adjacent motif, which is required for Cas9 endonuclease binding and cleavage. After binding to these PAM sequences, then only it can cleave the upstream target site. So here is no PAM sequences, therefore no binding and cleavage by endonuclease. But sometimes with even minor dissimilarities with the PAM sequences, the endonuclease Cas9 can bind, and in such cases the upstream of the PAM sequences may not always be the target site. So it causes the elimination of the other proteins apart from the target proteins. In addition, it is only possible for the non-essential proteins. With respect to essential proteins, the knockout cannot be executed as it leads to the cell death. So from all these discussions, what we can conclude is the wide research applications of the antibodies should be accompanied with proper validation for their specificity. Then it can lead to consistent results in the research. And while validating, it should be done via multiple tools and directions, that is, with appropriate positive and negative controls with the feasible techniques. These are the URL references of the images that I have used for making this presentation. I hope it is helpful for you friends. Thank you for watching this video.